leukocyte trafficking.
Marko Salmi, he is a professor of immunology, and Marko talks about trafficking, leukocyte trafficking, that has been his interest for 30 years or so. Please, Marko.
Thank you, and good afternoon to everyone. And now we'll switch back to a little bit more basic immunology and talk about leukocyte trafficking. So leukocytes are very mobile cells, and studying leukocyte trafficking has been one of the focus areas of immunological research in Turku for a long time, for obvious reasons. And it's very important that these different types of leukocytes scan our body constantly. Lymphocytes go from the blood to tissues, and then they come back to the blood and go to the next tissue. And in inflammation, both the lymphocytes and granulocytes need to leave the blood and enter the tissue to form the inflammatory reaction. And it's well known that this event can be misregulated in many cases, and it leads to different diseases, not only these autoimmune diseases, what Laura explained, and type 1 diabetes, but also in many other diseases, abnormal leukocyte migration plays an important role in the pathogenesis. And it's also, the field has advanced so much that there are already a couple of drugs on the market by which we can interfere the migration of the leukocytes in the body. And if we look at this leukocyte migration in more detail, there is a significant barrier for this migration, and that is the blood vessel wall. Here you can see that this is the inside of the blood vessel, then there is the endothelial layer of the vascular wall, and outside is the world where the inflammation takes place in tissues. And when these leukocytes leave the blood and enter the tissue, they need to take various steps on the endothelial cell to become arrested on the endothelium. Then they need to migrate through this endothelial barrier, and only in the tissue they can then form the inflammatory reaction. And these are the textbook molecules mediating leukocyte adhesion and trafficking in the body. But for different reasons, it was obvious a long time ago that this is not all. There has to be other types of molecules controlling leukocyte trafficking. And now I will take just one specific example to illustrate the different approaches we have taken here in Turku to study this leukocyte migration in detail. And that one molecule is vascular adhesion protein 1, or VAP1. And a very, very long time ago, we started to look for these new adhesion molecules by generating monoclonal antibodies against endothelial cells. And we found one which stained endothelial cells nicely and blocked lymphocyte adhesion to those cells. And those early times, it was necessary to clone the molecule to verify its identity, and it turned out that this is a very different type of adhesion molecule compared to the textbook molecules. And also the structure of the molecule was then resolved here in Turku. So this VAP1 molecule, also known as AOC3 by its gene name, it's an amine oxidase. It's an enzyme. It oxidatively deaminates primary amines in a reaction which then results in the production of the corresponding aldehyde, and at the same time ammonium and hydrogen peroxide is released. And that reaction takes place there deep inside the VAP1 molecule. And what we soon learned that this is really an enzyme which is on the cell surface, facing exterior of the endothelial cells. It was not specific for endothelial cells, it was also present on smooth muscle cells and adipocytes. but in inflammation on endothelial cell, it was very notably upregulated and located to the luminal surface of the vessel. And there it mediated the lymphocyte binding and allowed the lymphocytes to get into the tissues. So it took a lot of new assay development in which the flow, which mimics the blood vessel situation, was taken into account both in vitro and in vivo to really dissect the function of this molecule in leukocyte trafficking in detail. And now when we had an enzyme, it was an obvious question that what is the role of the enzymatic activity of this molecule, or are there other modes of action for VAP1 to mediate leukocyte migration? And we used this intravital video microscopy to formally prove how this molecule functions. We generated VAP1 deficient mice and compared the behavior of leukocytes in those animals to their control animals. And here you can see there is one vessel coming from the top to the bottom of the figure, and then there are some cells marked with red arrows, which are these firmly arrested cells, and some cells followed by the blue arrows, and they are the rolling cells. And in the wild type situation, you can see that there are lots of, this is a real-time image, video image, and you can see that there are lots of these slowly rolling cells within this venule. There are some which are firmly adhered to the vessel wall, and then there are leukocytes outside the vessel wall entering the inflammatory area. So this is the case in the normal mouse. And then when we compare the situation to the VAP1 deficient mice here on the right, you can see that these cells, they roll much faster than in the control mice. And the picture also looks cleaner. There are less of these adherent cells and extravasated cells. And when this all was quantified, it was evident that in the absence of VAP1, the leukocyte rolling velocity was much higher, while the numbers of adherent and transmigrated cells were lower. And that then resulted in a new concept in this leukocyte trafficking field, that stating that this kind of oxidative modification of the vessel wall can indeed contribute to the leukocyte migration process. And that led to a shift in some dogmas, that it's not only these traditional trafficking molecules which are involved here, but there is VAP1 and several other ectoenzymes which can modulate this process of inflammatory cell formation in a very strong manner. It stated here that the counterpart of VAP1 is unknown. What would be the substrate for the VAP1 molecule? And it took some years, but then two molecules, cyclic-9 and 10, were identified as counterreceptors for VAP1. And therefore, the current view is that VAP1 here on the endothelial surface is interacting with leukocytes using a very complex chain of events that the counterreceptors of VAP1 can bind to VAP1 molecule outside the enzymatic area of VAP1, and then also form an enzymatic reaction with VAP1. And these two phases of interaction are kind of separate, and both contribute to the adhesion of the leukocyte and transmigration of the leukocyte through the endothelium. And at the same time, this enzymatic reaction generates these end products, which are highly biologically active substances, and they further modulate the environment in which the extravasation takes place, and therefore they also contribute to the transmigration step. So now when we had identified a molecule which biologically mediates leukocyte trafficking, it was evident to test whether it plays a role in different inflammatory disease conditions. And here are some acute and chronic models that were set up here in Turku, and we showed that inhibition of VAP1 either by VAP1 targeting antibodies or by small molecule enzyme inhibitors which block the enzymatic activity, they can diminish inflammation in these conditions. And then this research has been spread throughout the world, and many other models have been used with different inflammatory diseases. And the observation is basically the same, that when you block VAP1 activity, the inflammatory infiltrate is reduced by 50% or so. And there is also a soluble form of VAP1 present in the blood. And it's upregulated in certain, but not all, inflammatory diseases. And especially we have done a lot of work in diabetes, in liver diseases, and then in cardiovascular diseases, and shown that the increased soluble VAP1 in serum is actually a good prognostic marker in those diseases. This is one example from the cardiovascular diseases in which all the traditional risk factors have been taken into account, and also the general inflammatory marker C-reactive protein. And in this model, the VAP1 concentration is still a very good or significant predictor of future cardiovascular event in currently healthy persons. It has the same prognostic value as the very widely used CRP determination. So it's something unique in VAP1 which is not just reflecting the overall inflammatory status, and it can be used in these disease entities to predict how the future of the patient is going to be. So then this basic immunological research has had another barrier, and that is to see whether any of these can be used clinically in any way. And during the years, there have been many antibodies against VAP1 which have been produced here, and they have been distributed to commercial companies, and the companies have also developed their own ELISAs for measuring soluble VAP1, and therefore the studies have expanded a lot at different parts of the world. These counter receptors of VAP1 have been utilized to generate new PET probes, which can be used to image inflammation in different parts of the body. And also, several companies have taken anti-VAP1 antibodies or inhibitors to clinical trials for treating different inflammatory conditions. And it all started here in Turku long time ago, and later on other small companies have generated their own VAP1 inhibitors, and some of them have been then acquired by bigger companies, and you can see that they have been tested in different indications, quite often in some liver diseases or then in diabetes. And one example of a very clear effect is this paper in which they found that inhibition of VAP1 alleviates diabetic kidney injury. So there are potential for using this molecular target in clinics as well. So this was just one example. There has been a lot of other activities regarding leukocyte trafficking in Turku, and therefore we have lots to offer for all collaborators, both academic and pharmaceutical ones. Nowadays, the studies have, of course, moved into the omics era and much more modern techniques than I showed here. But the important thing is that we have plenty of experience both with the blood vessels and the lymphatic vessels in understanding the molecules and mechanisms of leukocyte trafficking. And there are not only several other targets in addition to VAP1. Some of them are listed here, which we have found or been the first one to prove that they are involved in leukocyte trafficking. And the last one here is a molecule from which we are going to hear much more in detail in the next talk. So thank you, and if you have any questions, I can take them.
Okay, thank you. Are there questions? No? Are there questions? Yeah, Riitta?
If you inhibit VAP1, any side effects? And second question, what happens in infectious diseases?
Yeah.
So you will
repeat.
repeat. Okay,
Yeah.
you repeat.
So Rita asked whether there is any harm of inhibiting VAP1 and what is the relationship between VAP1 and different infectious diseases. And regarding the side effects, we know that long-term inhibition of VAP1 in animal models doesn't seem to cause any major side effects. And also the studies with the clinical trials have shown that it's safe and well tolerated to inhibit VAP1. In most cases, there may be some drug interactions with the small molecule inhibitors, but just as a biological target, it seems to be quite safe. That's probably due to the fact that it doesn't abolish leukocyte migration completely. It only reduces it. It shifts the balance to a direction that less cells go in, and that's useful to treat inflammation. In infectious diseases, VAP1 has been targeted or tested in several different microbial and viral infections, and there is a very small increase in the acute phase of the infection. So we can measure that there is more bacterial proliferation or viral proliferation at the early time points. But then again, compensatory mechanisms come in, and the end result is that the infections are not any more severe if the VAP1 is blocked. So it seems to be a very safe target for clinical trials with the current knowledge. Yes.
Jana? No? Not there either? No? Okay. Thank you very much.